Okay, so this is our pea glow transformation lab results. This first plate is LB negative, and it has a lot of growth because it is just E. coli by itself. Next, we have LB and ampicillin. This is the negative plate, and it has no growth because there was no plasmid DNA, and thus ampicillin killed all of the bacteria. Next, we have LB ampicillin plus, and this has growth, and it has two clusters of colonies, one with 102 colonies, the big one, and the smaller one with about 34 colonies. And this has growth because plasmid DNA was given to this plate, and plasmid DNA is beneficial genes for survival on ampicillin. Next, we have LB amp with arabinoids. This also has plasmid DNA, which is why it has growth. It has 6 small colonies, and the arabinoids makes it grow under UV light to show that it has transformed. It will be a bright green color. So the purpose for LB was to show the bacteria colony. So the purpose for LB negative was a control plate and to show how E. coli would grow without ampicillin, without anything, without the influence of plasmid DNA, and it's just E. coli by itself. The purpose of LB amp negative was another control plate, and is to show what ampicillin does to bacteria without plasmid DNA, which is, well, ampicillin kills it all. Next, we have LB ampicillin plus, and the purpose for this one was to show how the plasmid DNA is beneficial to bacteria by helping it to grow on ampicillin with the beneficial genes, which occurred because it grew while the one without plasmid DNA did not grow. And the last one we have is LB amp and arabinoids plus. And this one, the purpose for this plate was to show bacteria transformation under the UV light because it grows bright green under the UV. So there's going to be a picture of it right there. Yeah. Plus this one has ampicillin, and ampicillin lets it grow. Arabinoids is for it to glow, bright green, to show the transformation.
So in the SCD or the sickle cell disease, the patient will create these abnormal looking red blood cells instead of these normal ones, and this will block the patient into receiving blood to their vital organs. So scientists have just come up with this new therapy called the stem cell therapy, and basically what this does is that they will exert some bone marrow or these HS cells from their bone marrow, and then they will add some of these anti-sickling genes into the HS cells, and then they'll insert these cells back into the bone marrow, and then the bone marrow will instead make these normal looking cells instead of these sickle cells. And then usually these newborn normal looking cells will actually survive a lot more longer than the sickle cells, so the results are very long lasting. And one huge advantage of this method is that this can avoid the toxicity from the donor into the patient, and also it can prevent or avoid any type of patient refusal from any type of donor.
This is our Arabinose operon. It consists of 3 genes, ARAB, ARAA, and ARAD, and they code for 3 digestive enzymes. They involve the breakdown of arabinose, and they're clustered together. So these three proteins depend on initiation of transcription from a single promoter, which is PBAD. Transcription of these three genes requires a simultaneous presence of the DNA template promoter, RNA polymerase, and the DNA binding protein called ARAC. So, when arabinose is present in the environment, bacteria take it up. But once inside, the arabinose interacts directly with the ARAC, which is bound to the DNA, and this interaction causes ARAC to change shape, which in turn promotes the binding of RNA polymerase and the three genes ARAB, A, and D. They are transcribed. So three enzymes are produced, and they break down arabinose, and eventually the arabinose runs out. In the absence of arabinose, the RAC returns to its original shape and transcription is shut off.
So I'll be determining the transformation efficiency of the transformation log. So the transformation efficiency is a quantitative value that describes how effective you are at getting plasmid into the bacteria. So we are first going to determine the total amount of bacteria, and using the formula DNA in micrograms is equal to the concentration of DNA times the volume of DNA. So we got the numbers 0.08 and 10 from the packet. From the packet, in which they state that in this experiment you use 10 microliters of PGLO at concentration of 0.08 micrograms per microliter, and that is why that is the numbers we got, and we multiplied them together to get 0.8 micrograms. So this number will be used to multiply by the fraction of DNA used to determine the total amount of DNA spread in the agar plates, which was basically the LB, AMP, arabinose plates that we used. And for the next step, remember this is 0.08 is the total amount of DNA. So with that, we will determine the fraction of PGLO plasmid DNA spread into the LB, AMP, arabinose plate. Using the formula, the fraction of DNA used is equal to the volume of LB, AMP plate divided by the total volume on the test tube. So we got the numbers 10510 microliters from the packet, in which they state that you spread 100 microliters of cells containing DNA from a test tube containing total volume of 510 microliters of solution. So with that, we divide the two numbers together, 100 and 510. So the fraction of DNA used is equal to 0.1951, which is approximately 0.2. So this is the fraction of DNA used. So for the next step, we will determine how much DNA was spread in the arabinose plate using the formula PGLO. DNA spread in micrograms is equal to the total amount of DNA used in micrograms times the fraction of DNA used. So using our number from the very first part, which is determine the total amount of DNA, which is 0.8 micrograms. So we have the total amount of DNA, 0.8, times 0.2, which is the fraction of DNA used. Multiply them together to get PGLO DNA spread is equal to 0.6. However, if you're using the 0.1951, you would get the number 0.1568, which will then be rounded off to 0.16, which is about the same. Finally, we are going to determine the transformation efficiency. So the transformation efficiency, to determine it, you take the total number of colonies growing on the arabinose plate divided by the amount of DNA spread on the arabinose plate. So for our group, we got 6 colonies, and we take that number, which is the PGLO DNA spread of 0.16 micrograms, and divide them together. So 6 divided by 0.16 is equal to 37.5. However, if you're using a non-estimated value of 0.1568 instead of 0.16, you'll get 38.3, or about the same, if I'm correct. Yes, around 38. Now, for scientific notation, you basically just put it by a power of 10, multiply by 10. So we move the decimal 1 and we got 3.75 times 10 to the first power, which is basically the same as 37.5. So that is our scientific notation. And the transformation efficiency can be used to calculate how many transformant colonies would be expected to grow on the LB-AMP arabidopsis plate. So for our transformation efficiency, there are some factors that can affect it. So one of the factors would be the refrigeration of cultured E. coli plates. So there could be something wrong with the temperature or of the sort, because obviously 3.75 is a pretty low transformation efficiency. Therefore, there could have something that could have happened during the overnight process, or when we were doing the lab. Another factor which could affect the transformation efficiency would be the time on ice allowing the plasmid to come in contact with the cells. We could have possibly put too much ice on top or let it set in the ice for too long. In addition, we have heat shock treatment, it was not exactly 55 seconds, we went like 57 seconds. Factors affecting the transformation efficiency includes the diligence in going directly from the ice to 42 degrees back to the ice, which is the ice shock treatment to ice process. Another factor would be the amount of LB broth added, the recovery time in LB broth, the amount of cells spread on the plate, and the spreading transformants and controls. Now, the transformation efficiency, which of course for ours is pretty low compared to other groups, pretty much determines the quantitative value that describes how effective you are at getting plasmid into the bacteria. Since ours is low, we could determine that it was not as affected